For this talk, we're going to be discussing invasive blood pressure measurement and analysis. With the equipment and software, you're able to do both arterial and venous measurements and calculations. When I say measurements, most often our researchers are looking for sometimes as simple as heart rate measurements, maximum or minimum pressure, durations, sometimes getting as complex as doing isovolumetric time or tau. Oftentimes, our researchers want to take a look at these measurements in real time, meaning while the animal is anesthetized, they want to either add a drug or stimulate a nerve and see what happens with the blood pressure, how that affects the blood pressure calculations real time. These are called online calculations. So to show an example of that, this is the software that comes with iWorks equipment. It's called LabScribe. When we push the record button, you'll see a mouse blood pressure signal. So while this would be recorded while the mouse is anesthetized and the researcher is performing their experiment. So as I was saying, sometimes it's of interest for the researcher to want to do online measurements, meaning they want to keep track of certain parameters while the animal is anesthetized and they manipulate factors such as adding a drug or stimulating a nerve or a muscle. So we offer 8 online toolbars, they're called. Each of these toolbars will show a separate tab up on the top, which will follow different parameters, which means you're able to get or show parameters real time from 8 different sensors at one time. So when we click on one of those, again, it pulls up this top menu. If we go to setup of that top menu, we'll walk through how this shows. So we're able to set up that online calculation. First off, we're going to tell the software what channel it's looking at. So we're measuring from channel 1. We'll tell the software that we're measuring from channel 1. We could tell the software whether we want to average either cycles. So if you don't want to average, you'd put 1. If you wanted to average, so say that you want to take every 2 cycles and take the average of that, you can do that. Or you can average time-wise, say that you wanted every 20 seconds or every 30 seconds of data. To the right, the researcher here can determine what factors are important for them to monitor real time. If the beat number is important for them, whether the heart rate's important, the duration, the maximum pressure and minimum pressure. So they're able to go through and show as many factors as they would like and determine which ones are important and which ones they want to keep track of. Now, from that, iWorks actually has a built-in journal, so researchers can analyze the data right within LabScribe. If they want to export these numbers that are being generated, they can actually put that right into the journal, or they can export that to an Excel file or a TXT file right onto their desktop if they want to. From there, we're going to actually set the thresholds for detecting a cycle, which means we'll set these 2 blue lines and make sure that each one of these cycles actually sees both sides of these lines. Or if a researcher prefers, you can use the absolute values. When we click OK, we have our top menu that's been drawn up here. So now when we hit that record button, notice that the top numbers are actually changing in correlation with the recording. So again, the heart rate is changing real time with. So if a researcher or when a researcher wants to monitor real time changes to different stimuli, they're able to do that. The other type of calculation is offline calculation, which means the calculations are being done after the fact. So you've already had the animal on the operating table. You've done the procedure that you wanted to do, or maybe a researcher's looking for a knockout to see if a knockout variant has worked, then, or pressure volume loops, or however, whatever the researcher's looking to do with that. This will give you a data set. For example, here we have 2 arterial and 2 venous pressures. So there's 4 different channels being measured at one time. When we go into the advanced here, we can actually go to this offline calculations, which simply means we're looking at it after the data has been collected. So we'll click offline calculations. And again, we get that similar screen at the top. So again, the top channel here is our blood pressure channel from the recording. And we're able to tell it what blood pressure, what channel we want. We'll tell it that we are looking at arterial pressure. So your calculations are going to be specific, either arterial or ventricular pressure. Now, as we move to the next, we could have two other pressures if we want. We also have the ability to measure ECG. So again, if you wanted to look at end diastolic or determine end diastolic via ECG, you're able to tell the software to do that here and what channel to look at. Selection tab here will tell the software whether you're just looking at this selection of data, say that there's a small time in your data that you did a manipulation, say that you did a stimulation or you constricted something, but that will show you that small area of data. And that will show you those specific cycles. So in this small selection right here, we have 11 cycles. And as we increase that, our cycles increase here. Or we can actually take a look at the entire data file. So that will take the entire group of data from start to stop of recording. In our settings, we're telling the software now whether we want to use ECG for detecting that end diastolic. In our case, we didn't record ECG with this one, so we can use the derivative for detecting those cycles. We're also going to tell the software what the general rate we're looking for. So if you have a noisy trace, we're going to tell the software that anything that's below 10 beats per minute or anything above 1,000 beats per minute is going to be noised and to ignore it. We're going to set our cycle detection threshold sensitivity. So that's telling the software how sensitive, you're telling it how sensitive to be when looking for those. And again, we can smooth it. So say that we wanted to smooth that signal if it was a noisy signal. We can also adjust different parameters, again, for arterial and for ventricular pressures. So for arterial, we can take a look and determine what % recovery we're looking for. We can look at tau, relaxation time for ventricular, and we can determine if that's what we want those to be. So now we push the calculate button, and that will calculate those parameters for us. And again, as we increase or decrease, that's actually going to increase or decrease the number of cycles. So let's just put that at, say, 20 cycles. So we have 20 cycles shown here. We can click through and see each one of the cycle. The green trace is the cycle itself, so the cycle that's up here. The red trace is the average of those 20 cycles. So that's showing you how trace number 4 or cycle number 4 compares with the average. Now on our display right now, we're showing end systolic and end diastolic. We can also show systolic, diastolic, maximum dPdt, minimum, notch, which you're seeing right there anyway, or in the fill end. So it's up to you as to what you want to check or uncheck to show on those values. So again, we can go through and see how each one of those cycles relates to the rest of the data. Now on the bottom, again, we have a table here. On this table, we have first off, whether the selection is selected for up here, whether the cycle is selected. So say, let's take a look at cycle 4. We can click on cycle 4. It will show us the data for cycle 4. It will show us the trace for cycle 4. And it will show us that it's selected in here. So when we uncheck it, notice that our selection is now no longer selected. That's important because of those 20 cycles, we're able to get the mean value for heart rate, for example, the standard deviation, the maximum and the minimum. So say that we went through and we had an anomaly or an artifact, we can remove that from our data without having to go through and recalculate the data. Our algorithms were very clear with what we use and how the computer calculates, how the program calculates which heart rate and systolic index. So it's clear as to for the ES location. So it's clear which algorithm the computer and the software is using for each one of those parameters. Next, we have table options. So that again is showing you all the things that can be calculated. So every time that we had a researcher say that they might be interested in a certain parameter, it's been added to the software. So say that time is important. Again, you can click as to which ones are important for your particular study. So if it's not particularly important for your study or you're not as concerned with it, you don't have to show it. So let's show the max, min, mean. So again, we're able to go through and look at a variety of different parameters. So say that we were to go through and do all the parameters that we're able to determine from this. When we click OK, we have quite a table down here. So this table is easy to get copied. So we can copy and then just click and paste it right into an Excel or to a different format if we'd like. But that would make it easy for either for Excel if you wanted analysis that way, or for publication. It makes it easy for that as well. And again, you're able to see how many cycles you have, what's your mean for each of these parameters, what the standard deviation, the maximum, and the minimum. What we also allow is for that you can save the template. So say that you've gone through and you've made the table exactly how you want it to look for this particular study. You can save this template and that will save it so that the next time you can actually bring up that same template. Now with blood pressure, the features that make iWork specific to, and the features that are important for blood pressure is the ability to filter data to reduce noise. Obviously, you want a clean signal. That signal can either be straightened up in software with filters, or you have to control the noise around the animal and the surgery. Also, the ability to create multiple channels is going to be important for a researcher with that. So you're able to look at multiple different parameters, both arterial and ventricular pressures, and dedicated measurements for each. You're able to analyze either a complete or a part of a data file. So say that you were just interested in the part immediately before or immediately after a drug was added. You don't have to calculate, you don't have to take out that section and try to do analysis of that into a separate program. You're able to focus in right on that area that you're looking at. Or if you are interested in that whole animal, say it's a knockout, or say that you're looking at the overall effect of something, you're able to actually see over any period of time that way. The use of ECG to detect end diastolic is actually makes a much more accurate end diastolic as well. So that's you're able to do that.